Hi, good afternoon, and thanks for that introduction. And I was going to thank Alison for inviting me to talk, but haven't put me on after Alejandro. I'm not sure I'm going to thank her anymore. But anyway, one final comment on that. You've been watching, I guess, a talk on Star Trek. We're now back to Flash Gordon, black and white, so just be prepared. Now, of course, I didn't check how these things worked. Okay, so again, back to what are they? And I think Alessandro's slide has shown that very clearly. So they're chimeric antigen receptor T cells. So they're genetically engineered, and you can see now going forward they'll be multiply genetically engineered. And for now, autologous T cell receptors are what we're using. That's what we have licensed, what we have for use at St. James's at the moment. And as has been shown, they can be directed against any specified target antigen. For now, again, licensed and commercial, they're against the CD19 antigens, so we're using them in the context of relapsed and refractory diffuse large B-cell lymphomas, so aggressive B-cell lymphomas, and also in acute lymphoblastic leukemia, so B-cell acute lymphoblastic leukemia. But because their efficacy is not so good in the adults, they're only licensed in the for BALL in children and young adolescents. They're only licensed up to 24 years of age in that cohort. The one question I always ask is this a product, a drug, or a technology? And I guess from the European medicine agencies, they are licensed as a drug, despite the fact that they truly are a cell therapy. I think I've always struggled with that, really, but that's what it is, and that's what we have to live with. And I suppose the story of James is getting to the CAR T cell, well, we'll go back here for a second, getting to the CAR T cell therapy was that delay in getting funding for it. So it took us 3 years of negotiations with funding through the NCCP, through the HSE, and rightly so at the cost of it at one point. But I guess because it was licensed and because it had to come through a drugs panel, it was sort of harder to introduce. I think we could have snuck it through a lot quicker as a cell therapy if we'd been able to do that at the time. But that is the way the companies have gone with it, and probably been forced to go with it through the European Medicines Agency. Alejandro has again, most of these slides are now in black and white, as I said, from what you've seen before. But again, he's described that the, do I have a pointer on these? Okay, so what Alejandro's described is that the importance of this antigen recognition area here. So again, recognizing the antigen here on the tumor cell, which would be the CD19. What's also important, though, is this hinge region here, because this allows the flexibility of the CAR T cell therapy and allows it to get, I suppose, to awkward angles and to avoid steric hindrances. So while you mightn't think, gosh, well, that's just holding it in there, it has an important role to play. And then the importance of this co-stimulatory domain, but I'll just go into in a few minutes. Again, no point in having a CAR T cell with an activation if you don't have something that will continue, that will allow persistence and proliferation of the T cell. So this was the generations, and again, this is what this is describing. So we started off way back again with just a nondescript recognition domain and an intracellular CD3 zeta domain to activate the T cell. I mean, this work bound to something in a cell, was alive for a while, but then just died off. So of little clinical benefit. Here we got onto the single chain variable fragment lengths, which again we're now more specific, so we can imagine this being a CD19 cell. Again, CAR binds to the antigen, that's great, you get a response, but it activates, but again, they didn't proliferate, they didn't persist within the body, and again, of little clinical utility. And then we're on to where we are now, onto these third generation CAR T cells, and the difference here, as you can see now, is this co-stimulatory domain. So for now, the two products that we're using either use the CD28 or the 41BB domain. CD28 is good for activation, the 41BB is good for persistence in general, and I think we see that when we're using these products. But you can see, as Alessandro has talked to, by introducing multiple, getting that small train into a bigger train, now looking at putting the CD28 and the 41BB together, looking at putting the CD28, the 41BB, and all of these regulatory domains that you've alluded to, the interleukin-2, the interleukin-15, things that will maybe allow better penetration into solid tumors. But all of them will need a CD3 zeta domain, which is here, which will activate the T cell and get that response that you need. For now, we're working in here, and for now, really, these products are only working in blood tumors or in blood cancers. And you can imagine that these cells are circulating. These CAR T cells are injected in, so they're circulating, so they're meeting each other and knocking each other out, and that's working well. These aren't working well in solid in solid tumours. And the reason for that, again, is that as you can imagine, a solid tumour, it's in a what's called a hostile microenvironment. So you can get the CAR T cell to it, but if you don't mind me using your Mexican police, they can look in, but they can't get through and get in there. And that's the idea of adding all of these extra domains, is to allow that penetration into the tissue tumour. So what they're doing with products like this now is that if you can directly inject that into the tumour lesion, then you will get some efficacy. So we're seeing that now in clinical trials coming from San Francisco and glioblastoma, that injecting directly into the ventricular system and just delivering that CAR T to the tumour, you're getting cell death. That's requiring multiple tumour infusions. I see multiple infusions of therapy, and at 300,000 a dose, it's probably for the rich Real Housewives of Beverly Hills and not for the James Street cohort. Other thing, again, as I think you mentioned there, the HER2 and whatever, again, trying to use these in breast cancer and getting some efficacy, but again, very difficult to get those T cells to persist and act. Again, gone through this, but again, this whole cycle of getting from the patient, getting this processing here, sending the cells away, getting them frozen, delivery. I think that's something, I suppose, Thomas St. James the first year that the lab have found, this takes time. It sounds like, okay, the cells just come to the lab, we'll just send them out. But actually, there's a lot of paperwork around that cells. For some of the companies, you have to freeze the cells. For other companies, they'll pick them up fresh. It takes a lot of time. There's a lot of document checking, and I think maybe it's something we underestimated the time that would take. And Nicola's nodding here with me while we set this program up. But at least the cells are picked up, they're gone away. They come back 28 days later. So that's been about our average delivery time, 25 to 28 days. Patient comes in 3 days prior to cell reinfusion for some lymphodepletion therapy, and the CAR T cells are reinfused. And it's at this point that the pharmacists are involved here, that when the cell comes back, the pharmacy, for the first time probably ever, for you to release a cell, to release a product from your lab and from the cryobiology lab, they have to deal with the pharmacy who come and will check the paperwork. And as you said, it's quite a laborious process. What we are learning is that this 28 days is not necessary. It takes about 4 or 5 days to actually make the CAR T cell. Then we were led to believe, well, I was led to believe and thought that the next 20 days were all to do with quality processing and getting quality out. But now I'm hearing that, well, really it only takes a couple of days for that quality processing. It's just that the companies have so limited slots and so few people. And so I think that it's taken 21 days to do a 5-day QP release. And that's where, again, doing it yourself, getting the cells, if you can generate your own CAR T cell at home, you're likely to get a vein-to-vein time of 7 days, which is hugely important for these patients with aggressive tumours. And you'll know, any of you who are following that literature, that in the initial clinical trials, that about a third of patients died while waiting in that 28 days for their CAR T cells to arrive. Now, at that time, patients weren't allowed to have bridging therapy, so they had to sit without therapy for an aggressive lymphoma, which I think as clinicians, very few of us would sit on anyway. But nowadays, we can use bridging therapy while waiting to deliver the, get the CAR T cells. Probably don't need to go through this in any great detail, but again, not an overnight sensation. People have been thinking about T cells and tumours for a long time. And as Paul said, in 2001, 2003, I was an immunology SPR, and I remember going to a meeting in the UK on an immunology meeting. And I think even at that time, not everybody believed that the immune system was even related to cancers, in fairness, or that there was anything. And I remember specifically that talk, and I remember the immunologist saying, sure, Jesus, what are these cancer doctors doing even dealing with this? They're using IgG4 antibodies when they should be using IgG1 antibodies, et cetera, et cetera. So it is interesting that we're now gone 20 years later, and we've absolutely determined that the immune system is central to development of cancers. And by honing in that immune system and harnessing that immune system, we can maybe get better therapies. But as you can see, for 2003, there was a CD19 CAR T. 2013, the first clinical trial, that first patient had CAR T. And we had the pleasure of having Emily Whitehead over here but 6 months ago. So the first patient ever to receive, first child to receive a CAR T cell therapy for acute lymphoblastic leukemia. Herself and her dad and mum came, gave an incredible story of driving home on her child who had failed her 4th line of chemotherapy, was not eligible for a stem cell transplant, was heading home essentially for palliative care and to die. Phone call made to Children's Hospital Philadelphia, and they had just opened their first CAR T trial the day before. So they turned the car around, drove to Philadelphia. That's 11 years ago, and their child got their first CAR T cell therapy ever given to a child. She spent, I think, 10 days in ICU, nearly died from cytokine storm. They didn't know how to treat it. eventually somebody in the lab found IL-6 was high, and they found tocilizumab and they gave it to the patient. This is the dad's story. They had been told that she would die. They wanted to turn off the machines. He said, give me one more night. That's when the IL-6, the tocilizumab arrived. And according to him, the child woke up the next morning. It's the story he gives, and that child was with him, now 16 years of age, perfectly well, and heading off to college. So it's a phenomenal story. So where are we now? So we have, from the EMA, 4 products approved. So we have Tithogen occlusal from Novartis, and we have Axicel, Axicaptogene Quellolucil from Gilead and Kite. We have these companies, or these products now available in Ireland for therapy. We're now at 2022. You can see these have been approved back in June 2018, which shows the difficulty in getting these expensive therapies through. And should we have a therapy that's 10,000 euros, hopefully that's not going to be a problem going forward. Tecartus is approved since 2020. We still do not have a license or approval for funding for Tecartus. And then Isocaptogene, which is a BMS and Juno product, is not, the company haven't even started in Europe to try and get this company. So we're at the fate of what the companies want to do, where they want to sell them, and when they want to sell them. The interesting thing, so these all look quite similar, so where are the products? So the Isocaptogene, what they've done differently, or what BMS have done differently with their CAR T cells, is that instead of just taking the T cells, and starting that viral process, they're now pre-selecting the T cells and ensuring the CD4 to CD8 ratio is 1-to-1. I don't know that they'll know that's any better or not, but actually in the clinical trials it seems to be better. It seems to be more efficacious and less toxic. As I said, for now this is not even on Celgene's mind to even bring to Ireland, so this is not going to be here. Tecartus, I think, is in discussion. We haven't had a requirement for mantle cell. Those of you who've had patients and have been dealing with us for the last 3 years will know that while waiting for this to be funded, we did have permission to send these patients to the UK. So thanks to colleagues in King's and in UCL, we were able to send patients for CAR T cell therapy. We've sent 3 patients for mantle cell therapy to date. We haven't had a requirement to send a patient with mantle cells to the UK since we've had these products available here in Ireland in the past year, and we wonder, will we be asked to give the mantle cell product when we start looking for funding to send it to the UK? So we'll wait and see what happens there. Exciting to those of you in the room too are now that the EMA have approved 2 products for multiple myeloma. So we have the BMS Abecma project and the Carvykti from Janssen. We are hoping in the next month to open a clinical trial with Janssen, this CARTITUDE-5 study, which will look at treating patients with multiple myeloma as a frontline therapy. It's for patients the wording is unsuitable for autologous stem cell transplant. The word there meaning that it doesn't say ineligible. Or not, I didn't even say unsuitable, it says not going for autologous stem cell transplant. There's been an update on that wording, but for now it gave us some potential to use these products. So I'm just going to go through some of the clinical data for you fairly quickly. And I'm going to, and what Alejandro had alluded to in bone marrow transplant is to remember that these curves for patients who wouldn't have had these therapies, we're down at the 10%. So the big scholar study, which looked at all of the outcomes for patients with refractory lymphoma in over a series of 3 huge trials in the UK and Europe, showed that the overall survival was expected to be about 7 to 10% at 1 year. So while these numbers may look a bit low down here at the 40%, it's 4 times better than the best available therapy up until these were available. So Novartis Tithogen in the Eliana trials for BLL showed good survivals. Again, we're looking here at that young adolescent cohort up to age 24. The older patient, those curves were heading back down towards the 20s. And for now, that hasn't, we don't have approval for use of them in the older adult, except now the Gilead Kite people who probably have a slightly better product for ALL have got approval in the US. We don't have it in Europe yet. And again, this is the trials with lymphoma, again showing pretty good overall survivals. Again, this is for all patients, but for those patients who have a complete response at month 1, you can see survivals are up as high as 70%. So again, compare that to 7%, you take your chances if you're offered this therapy, I think, going forward. Axicaptogene, again, from the other company, from Gilead Kite. Again, a similar pattern. There's probably no need to go into the details of that. But the key thing is get the therapy, get the response in the first month. You're about a 70% chance of an overall survival. These are going up to 2 years. I think real-world data probably will come back up a NASH now, which will get us to 3 years of those therapies. So I haven't seen the abstract yet to see what's going on there. And then finally, this is a... This is an amazing product, really. When you look at it, this is the Janssen product for multiple myeloma. You can see maybe similar curves, but you've got to look here. Look at this, a median of 6 prior treatment regimens. So absolutely phenomenal results for this BCMA product. So I think this probably will change the way patients with myeloma are treated. Again, you know, looking at costs and everything that have to be taken into account, but pretty outstanding results. Caveat I'd add to it, and not to rain on the parade, is that myeloma, I guess, is a chronic disease relative to an acute lymphoma or acute lymphoblastic leukemia. So I guess you'd like to see 5-year survivals of these to be sure that we're maintaining that response, but it is looking pretty good. I think if you told anybody that before CAR-T, after 6 lines of therapy, there's a 70% chance of being alive for 2 years, I don't think anybody would have ever said that to a patient, in fairness. So outstanding results so far with the Janssen product. So hopefully they'll come in at a cost that's you know, that won't delay implementation and availability for all the amount of patients with multiple myeloma. The target antigen, as I said, is the BCMA thing. Well, I'll talk to it in a second. Safety profile. So again, like I think I remember from pharmacology lectures, there's no good drug that doesn't have a good side effect. So side effects are there, and the big things, as we said, are this cytokine release syndrome. So that acute inflammatory reaction you get when you reintroduce T cells into a host, and we really want to see that happening, really, we think, because you want to see that's the marker of the T cell expansion that's happening, and the bigger that T cell expansion. Certainly in my practice, the safer I feel that maybe this product is going to work. The worst feeling is watching that patient for the week on the ward with no temperature and no fever, feeling perfectly well, and then even the patient, because I've warned them about the CRS, starts to get worried that they're not getting the temperature. So it's a double whammy. So cytokine release syndrome here, in about 12% of patients. I think that's probably about right, grade 3. So life has changed since these were the original clinical trials where patients investigators would have been, you know, almost afraid to go in and try and abrogate that CRS response because they wasn't sure what the effect would be on the actual function of the CAR T cell therapy. I suppose if anything that COVID brought, it did bring a fear in the UK of patients going to the ICU because they knew they didn't have capacity in ICU. So what they did was they started to treat that cytokine release at grade 1 or grade 2 levels, which was much earlier than they would, but they couldn't afford the chance that that patient would need an ICU and would not get a bed. But it did not seem to impact outcomes for patients. So we initially, when we were gingerly went into this, we opened the program last December. We gingerly went into it. So patients with a fever that lasted for about 24, 36 hours, even if they were well, we did go in with tocilizumab. We were probably treating ourselves and doing a disservice to the patient. Now, as time's gone on, unfortunately, those patients are left withering on the ward for 3 or 4 days with parexias of 39 and 40 before we'd even consider giving them anything to abrogate the response. So that's the CRS. And then the second thing that's unique to CAR T cell therapy is the ICANS, the immune effector cell associated neurotoxicity syndrome. It used to be called CRES, which was much simpler, but in encephalopathy syndrome. Again, you see here up to 30% of patients, but again, in clinical trial situation where patients were, there was a fear of treating that patient. Again, you didn't want to kill off the CAR T cell. So we go in quite early if you start seeing neurological syndromes, and we've only had, out of the 27 patients we've done, only had 25 patients we've done, only had 2 patients with encephalopathy syndromes requiring transfer to ICU. Both of those patients only had about a 2-day stay in ICU, and nobody died, thankfully. So what's the future? I think the future's just been told to us. I won't say too much about it. The armored cars, which is what Alejandro has talked about, the Sleeping Beauty system there. Dual antigenic targets, again, it's all part of that. CARs are now triple antigenic targets are there, and again, off-the-shelf allogeneic CAR Ts. So again, there's no need for me to go into them. You've heard all about them. Multiple infusions, again, is it better just to keep adding in? First trials have suggested maybe not, but it seems like if you're directly if you're directly delivering to the actual tumour, that maybe multiple infusions is the way to go. And then the combination therapy with PD-1 inhibitors. So that's where you would use a PD-1 inhibitor to open up the CAR. So cancer cells can hide behind their PD-1, or they express a PD-1 to stop the T cells getting in there. So maybe the reason that some of the CAR Ts are working is because they can't get into the tumour. So by using a combined PD-1 inhibitor, that may help that. Again, the disease targets, we talked about the solid cancers, this insufficient anti-tumor activity, this poor persistent tumor site. And Alejandro's just left now, but he nearly didn't mention benign diseases until his last bloody slide when he put in autoimmune diseases at the bottom. But there is CD19 targeted T cells now in SLE and systemic lupus erythematosus. So the first publication, I think, of a non-cancer diagnosis in humans. So then this came out in New Scientist Health on the 15th of September of this year, showing that CAR T cell therapy in, I think, 4 patients with lupus showing very effective results initially. Anyway, again, it's a chronic disease, you have to wait and see what happens, but it did prompt our immunologist to ask us when we were starting to do CAR T for SLE. So it's good news. Sometimes you have to be conscious of what impact it's going to have on a service. And so for us at St. James's, we're ongoing with our developing clinical program. So as I said, in the first year we expected 24 patients. We thought 2 a month. What happens, we've done 26 now in about a year, so we're probably on target, and there's Nicola probably had the details of the number of patients that we have collected, but we've about 4 extra collected that we haven't reinfused yet. And then we have on the basis of this CAR T cell therapy and other cellular therapies that are happening with our colleague Dr. O'Fally as well in St. James's, and with Nicola and Derek Doherty, have set up a cellular therapy research group. I think Nicola's going to talk to you in more detail about that in the next 20 minutes. And so there you go. So I got through it anyway, and most people listened after that phenomenal talk beforehand. But we have a novel therapy for poor prognosis diseases, although small phase 2 trials would seem to be holding well up in real world data to now. Short follow-up, but we'll wait and see what happens. And as you can see, the indications are increasing. So thank you very much for your attention.
Well, thank you very much, Larry, and it's a fantastic overview of what's actually been delivered, and I think you're rather modest, but you do deserve personally congratulations from all of us on bringing this program into Ireland. It's hard to overstate the amount of behind-the-scenes work that's involved in persuading so many stakeholders, if that's the right word. But, you know, and I think it's been an extraordinary achievement, and it's fantastic having worked with you over the last year, particularly over the last 9 months, seeing these patients and trying to help look after them. It's been an extraordinary experience, and as we've just heard, it's only beginning. So we have time for a couple of questions for Prof. Bacon, or comments or clarifications. Derek.
Yeah, I think we're definitely, recognizing the tumor antigen is less of a problem than recognizing the off-tumor effect of that antigen. So I think that's where the difficulty will lie, and I think even in the autoimmune conditions, I think at a recent talk somebody asked about ulcerative colitis again, and again, if you can define that antigen that has a non-treatment and an off-tumor effect, and no off-target effect, I should say, or no off-tumor effect and no target, you know what I'm saying, that it's limitless, really, I think, to be honest with you, because the CAR T cell therapy itself, from what we're seeing, and now I have to admit, we're delivering it in an environment which is used to stem cell transplantation, allogeneic transplantation, used to sick patients, used to, preempting what's going to happen. But in that environment, it's a very safe therapy. It's a once-off therapy, potentially. It's a 2-week hospital stay. And the follow-up so far, is not particularly there's not a huge amount of toxicity afterwards that requires a lot of resource, I would say. So in truth, I would imagine it's limitless, and I think you can see that from what they're going to do, what potentially can be done with these. But the limit will be is identifying the target that is tumor specific, and I think other than that, I think it can open to any range of autoimmune or immune mediated diseases. Not sure how it'll work in the Aspergillus itself, but yes, so there you go. Even in infections, well, we'll see.
Other questions or comments right now? Well, as I say, anybody who's interested in joining the program that Larry and colleagues have put together, or contributing in any way, or working, and again, very grateful to the colleagues in the IBTS, many of whom are here, for the support, but just contact Larry or Nicola directly. Thank you, Larry.
Yep, love it, thank you.